pleasure to welcome Dr. Robert Naczi to our stage. Dr. Naczi is going to tell us more about the Taft NYBG Internship Program. He is the curator of North American Botany at the New York Botanical Garden, and since its inception 9 years ago, he has coordinated the Taft Internship Program and has also served as a mentor for a handful of students, including myself. Please welcome Dr. Robert Naczi.
Good morning, everyone. And Rachel, thank you very much for that kind introduction. It's very exciting for me to be here because, as Rachel mentioned, I'm talking about this very special partnership between Taft and New York Botanical Garden. And so I will take the next few minutes to tell you about this. And this is truly, you see some friends here. Truly, this is one of the best partnerships. And that's because it involves you, students from Taft School, and us scientists from New York Botanical Garden. It's also the best partnership because it's mutually beneficial. This is truly a win-win situation. So, as Rachel mentioned, during the last years, we've had several interns, 10 to be exact, and you hopefully will recognize friends among these. There are many, many benefits because by now you're thinking, what's in it for me? That's a good question, and you should ask that. So first of all, an obvious one, and that is the experience. Participating in cutting-edge research. This is original research that we propose every year from different curators at the garden. Not only the experience, but also being part of a community of scholars. So these are just a few of the interns in the capstone experience, the intern symposium at the end of last summer. So we typically have 20 to 30 interns, and these interns range from high school through undergraduates to graduates to even postdoctoral scholars. So it's a wonderful opportunity to engage with other like-minded young scientists. In fact, we have a weekly internship enrichment program of which this capstone experience is the ultimate. Another thing is that we have a very exciting summer planned at New York Botanical Garden. So we have our conservatory with its many glass houses, each one simulating different natural habitats throughout the world. We also have our various outdoor gardens, such as the azalea garden, the conifer arboretum, and the rose garden. At the core of the garden is the Thane family forest. This is a 50-acre natural area, and in the center of it is a tract of old-growth forest, one of the 2 remaining in New York City. We also have an engaging lecture series. We have more than one, in fact. We have one in science, and then we have special lectures in horticulture throughout the summer. And then this summer, we're excited that we will be hosting an exhibition of the art of Yayoi Kusama, and that will be running through the season. Now, in addition to the background activities of the garden, we also have, of course, science. And in science at New York Botanical Garden, we have a lot to offer. First and foremost, and dearest to my heart, is the herbarium. The herbarium is a museum of plant specimens collected from throughout the world going way back to the 1600s. This is the largest herbarium in the Western Hemisphere with over 7 million specimens. We also have our Geographic Information Systems Laboratory, which allows us curators to explore geographic data from our collections. We also have the Molecular Systematics and Structural Botany Laboratory off in the Pfizer Plant Research Laboratory, a separate building. In terms of the areas of expertise among us in science at the garden, first and foremost, we have systematics. This is the branch of biology that concerns the identification and relationships among organisms, in this case, plants. We also have floristics, the study of the distributions and frequency and geography of plants throughout the world. Most of us curators have an area of systematics expertise, but also floristics expertise. So we work in various parts of the world. We also have ethnobotany, that branch of botany that concerns human relations with plants, especially uses of plants, such as medicinals and poisonous plants, and timber and non-timber forest products. Now, I wouldn't be giving you the full story if I didn't tell you, as I mentioned before, this is mutually beneficial, so there's something in it for us too. We, the curators, benefit because we get to work with you. And I say that because we're all truly excited to engage with young people. All of us are educators. Every one of us has at least one adjunct appointment with nearby universities, for example. But we also like to work with high school students. We like to work with you because we want to help forge the next generation of scientists. And if you become a botanist along the way, that's all the better. We also receive help from you. So as I mentioned, we conduct real research projects. All of us are very busy, and we benefit from your talents and expertise. This is an intensive program. We expect that all interns will treat this as a full-time opportunity for the summer. Now at this point, I'd like to wrap up by quickly introducing just a few of the curators and their specialties, so you have an idea of the wide-ranging palette of experiences that you could gain at the garden. First is Barbara Ambrose. She is a developmental botanist, and she uses techniques of genomics to study the evolution of lycophytes and ferns. Andrew Henderson is someone who specializes on the systematics and geography of Asian palms. Lately, he has been working very actively in Vietnam and has discovered over 30 new species of palms. And remember, palms are large and showy, so this is quite impressive. I work on the systematics and floristics of North America, particularly the northeastern quadrant of the continent. And I'm working on this book, The New Manual of Vascular Plants of Northeastern United States and Adjacent Canada. Matthew Pace works on the orchid family, and he's especially interested in cryptic speciation. Like Andrew Henderson with his palms, but at a much smaller scale, Matthew is discovering loads of new species as he looks deeply, especially using techniques of molecular biology to elucidate relationships among these orchids. Greg Plunkett studies the carrot family. He's very interested in the systematics, and he's looking at large, diverse lineages of these carrot plants, especially in South America. And Ina van der Broek is an ethnobotanist, as you can see, and she's especially interested in traditional knowledge from the Caribbean, in situ, but also in New York City, and how those cultural traditions change and are adapted when they're brought into the urban environment. At this point, I'd like to acknowledge many, many people who have been very generous with us, and I'll highlight just a few. You can see the rest of them. Scott Morai and Paul Parvist are the ones who actually had the genius to create this internship program, and I owe a great debt of gratitude to both of them. We also have benefited from many benefactors in the Taft community, and I'd like to highlight Dwight Stocker. Dwight is in the audience here, and Dwight, go ahead and wave if you'd like. Dwight has been very generous. So I'm the one on the New York Botanical Gardens side who coordinates the internships, but Mrs. Guidati is a very able and very friendly and engaging, of course, you all know that, partner here at Taft. We would not be able to do this without her. And of course, this is all possible only because of the support of the Taft School. So I thank Headmaster McMullen, Chris Latham, Lawrence Crimmins from the Development Department, and the entire Taft Science Department. So with that, I would like to introduce Rachel Peverly, who will tell you a bit about her experience from last summer. Thank you.
Yes, Rachel! Thanks, hi. So for those of you who don't know me, I'm Rachel. I'm a 3-year senior, and I'll be talking about my experience interning at the garden this summer. Next slide, please. Thank you. Okay, so as you guys know, I had the privilege of being mentored by Dr. Naczi, and I worked on a project that would be put towards the new manual of vascular plants. Essentially, I worked on a proof of concept for what can be summarized as an identification manual for both researchers and beginners, the common public. And I put together the species description, but more specifically, I worked on constructing different visual aids to help identify the species. All right, so a couple of the resources that I used, Dr. Naczi did talk about. I was in the herbarium quite a bit. I handled hundreds of species looking for a representative model that could be used for about 45, 46 species of the genus Cyperus. And yeah, so it was really cool. I got to get to know the herbarium. I got to understand how it was organized. And it was just a really cool experience to handle all these rare and really old documents, different specimens. Additionally, oh, sorry, you can go back. Thank you. Additionally, I also worked with this high-resolution microscope. It's similar to the dissection scopes that we have in WU, except it's hooked up to a computer. And that computer puts the image onto a screen so that you can create like pictures, and we use stacking software to make a 3D image. And so we're working with really, really small characteristics of the plant, and so just to capture these in really fine detail was a really cool opportunity, really great experience to handle a scope like that. So. And then Dr. Naczi talked a little bit about these other opportunities that the interns have, but I'll just go in depth a little bit further. The Richmond activities are different every week, and they're always really, really interesting. It's a great way to meet different researchers at the garden. I know one week we toured the conservatory, which is probably the most famous of the garden buildings. And additionally, there was another week that we heard a lecture from someone who works a lot in the Amazon. So we got to understand his experience working there, and and also another time we were looking through the rare books room in the library. These books are hundreds of years old, so just to be able to learn a bit about them and learn a bit about their history was a really cool opportunity. And there's also a symposium, as Dr. Naczi mentioned earlier. It's a great way to build your presentation skills. If you're looking to go into science, being able to convey what you've discovered or what you're working on to an audience that doesn't necessarily know what you're talking about is really important. Also, scientific presentations are a little bit different than the presentations that we do in the classroom. So just getting tips from professionals was a really awesome experience. Working with different interns was really, really amazing. A lot of these interns are graduate students or juniors and seniors in undergraduate programs. So Eugene and I were really lucky to be one of the few high schoolers that get this opportunity. So I'd just like to thank Dr. Naczi first for being my mentor, for sharing his insight into the world of botany, his experience inside the garden and his external research. Additionally, I would like to thank Donald B. Stott for supporting my internship. It was a long commute, but it was definitely worth it, so everything helped. And I also just want to reiterate that the partnership that Taft and the New York Botanical Gardens is so incredible. It's such an incredible opportunity for people our age to get hands-on experience in the lab. So with that, if you're interested, please feel free to contact me. I love to talk about it. If you have any questions, I'll help you get in touch with people, but please consider interning at the New York Botanical Garden. Thank you.
Good morning, everyone. My name is Eugene Acevedo, and this summer I had the privilege to intern at the New York Botanical Garden and shadow Dr. Damon Little. This all started when I was looking for something interesting to do over the summer. Next slide. My initial idea was to spend days on end at the pool, and all I was saying in February was, summer finna be a movie. When I actually put things into perspective, I realized that doing something productive will most likely benefit me in the long run, and I will actually do something productive over the summer. I will not only gain some work experience, but also try something new. Next slide. To start off, the overall goal was to relate the cell measurements to environmental parameters, such as the soil data and climate data. This would ultimately allow me to see where exactly the conifer is found. To get to this goal, my job included preparing certain samples, specifically conifers for the scanning electron microscope. Next slide. Sample preparation included 6 steps. First one being collection of samples, second, slicing the samples in half, third, fixation, 4, critical point drying, 5, mounting using adhesives, and lastly, 6, butter coating. Next slide. Each of the steps were very challenging, especially slicing the samples in half with the razor. It was tough, but I got through it. The razors were very small, and that was me. I was sad. The point of this was in order to fully see the vascular system, which deals with the transportation of water, Next step. Next slide. The next step was to remove wax and dehydrate the sample. In order to do this, a subtle increase of ethanol every 2 hours was required, in addition to the use of acetone at the very end. Next slide. The last 2 steps were mounting and sputter coating, which in my opinion were the most essential steps, as they are the reason why the scanning electron microscope is able to work and take images. The sputter coating makes the samples conductive and allows the electrons to bounce off of it and produce an image, which you're going to see later on. Next slide. This is the stew. That's what I called it. This where I was take images. The left is the actual where I would actually put the studs in. TV where I would see where I was looking for the transfusion tracheids. Next slide. This is a picture of your boy. I was measuring tracheids, cell width, cell area, and counting the number of pits. I would spend days on end just measuring. That's me. Next slide. These are the few images that I took on the scanning electron microscope. Kind of dope, in my opinion. Next slide. This is another image that I took. Some of the images did not come out like this. Some were messed up, like not very clear, but a few came out very nicely. Next slide. After measuring the cell width and the thickening length, we graphed them, and we're able to use a program to find where exactly in California it was found, which was approximately north of California. Next slide. In conclusion, I would like to thank everyone who made this happen: the Taft School, Ms. Fry, Ms. Guidati, Mr. McAloon, Dr. Parvis, Dr. Damon Little, the New York Botanical Garden, Mr. Dwight Stalker, and the Big Foundation. If you have any questions, do not hesitate to stop me in the hallways. Do not hesitate to ask questions to Ms. Guidati. That's it. Thank you.